So I'm here in Seattle, and I'm joined by Patrina Pellet. She is at the beautiful location at the MBL in Woods Hole on the other coast, on the East Coast. And today we're going to be talking about science communication for microscopists or for microscopy, depending on how you see it. We'll have a brief introduction, which is then followed by Patrina giving very valuable pointers on image acquisition, and then she will go through a case study, an example of how she prepares data for presentation, which is very valuable. I'll then talk a little bit about how to do volume and surface rendering, and then preparing data using video animations, augmented reality, or virtual reality to present data, depending on what kind of media you're presenting on. And then a few conclusions, and we have, of course, time for questions and answers at the end. And we expect this webinar to be approximately 45 minutes from me and Patrina, and then maybe 15 minutes or more if needed for questions and answers, and maybe some more examples using Aivia to illustrate. So communication of science hasn't started with Twitter. It has started way before, and the earliest reference I could find is actually pretty old, 30,000 years before the birth of Jesus, so BC, and that is when essentially people were painting on caves and telling stories. And they used this way to this way of transferring information. And some of the information was scientific in nature, even though science wasn't yet invented. Some of these paintings depict either ways to capture animals and eat them, or migration patterns. And so you could consider this a little bit of a science, or proto-science as it's called. So the earliest paintings that are known are around 30,000 years, 32,000 years old. Then in terms of the next stage would be books. And the earliest books one can find are clay tablets. And this particular clay tablet is almost 3,500 years. It's dated 3,500 years BC. And it has information about how the stars, the moon, and some planets are thought to move. And that's pretty remarkable, considering there were very few tools at the time to actually observe the motions. But even with the naked eye, they could already make some observations, and then they recorded them in these very beautiful tablets. This particular one is kept at the Louvre in France, in Paris. So fast forward quite a few centuries. In 1665, this is AC now, is the first publication using what we call now peer-reviewed science publications. In fact, early on, this first science journal wasn't peer-reviewed, but pretty quickly the peer review process was put in place. So within 20 or 30 years, there was already a sort of peer review process in place. But the earliest ones didn't have any. It was they were more of a record of experiments and some opinions. So it wasn't exactly as it is today, shaped, but it was very similar. And actually, this is the first example of a scientific journal. Nowadays, we use a lot. We disseminate our science through scientific meetings, and there are, of course, museums. And more recently, the last 30, 40 years, the internet has really made it possible to put out information at a much earlier stage and with a much higher frequency. So at least the bandwidth, the amount of data that is available for the public has increased significantly. And that's definitely a good thing for everybody to not only understand what science is all about, but get better informed and make better decisions. If we think about microscopy specifically, microscopy started, or the microscopy that we know about, started and we look at is using either a single lens or a compound microscope. So the compound microscope is thought to have been invented in the 1500s, but it was in the 1600s that we have the first recording of an image that was captured by a microscopist that was looking at bees, and he recorded this drawing. He made this drawing. This is the first illustration enabled by microscopy. That's the first that we could find. I don't think there's anything older than this. And this is from 1630, and it's using a Galileo microscope, compound microscope. There were many other key researchers or key microscopists along the way, and some of them are here. And this is just the let's say the old wave of microscopy. Now in the 20th century, I'm not covering it here, but up to the 19th century, you can see there are quite a few steps. But this is the first illustration enabled by microscopy. So illustrations were always a key way to show what you could see on the microscope. And since it wasn't possible to create images with film or digital images like we create today, then the scientists, microscopists, did it by hand. Today, however, things have progressed a lot, and really the internet has become the key way to communicate science. Even the scientific publications, most of them are read through a web browser, not really in paper format. But we'll get there in a moment. One of the key things that has changed in the last 5 to 10 years is Twitter, and there is an enormous amount of data being put on Twitter and shared there, and it's used really as a way to attract awareness to a new publication or to a new research group, or just to let people know what it is like to be a scientist. And there's a few interesting hashtags that people follow, like actual living scientist and others similar to that give the wider public a sense of what it is like to be a scientist. So Twitter has been a great force, and we use it as well. And I know Patrina uses it as well, not just to talk about science, but to talk about other things as well. And the immediateness of it is something that is very, very attractive. On the other end of the scale, so the Twitter, you could say it's mostly about fun and getting a word out quickly. On the other end is actual research work being presented in a classical format of a journal is bioarchives or preprints in general, but bioarchive is the most popular one in our space. This really took off. Was only set up in 2014, but you can see here, this plot shows how quickly it has grown versus the more standard ways of publishing. And that, I think, is going to continue because there are quite a few things that are very attractive about bioarchives. And it kind of turns the whole process of peer review upside down. And that's very attractive to the younger scientists that are keen to get their work out quickly and get comments from potential colleagues or collaborators. And in many cases, if the publication is good or the preprint is good, it gets picked up by a publisher and eventually gets published in a magazine too, a conventional magazine. But it has grown tremendously. On both of these and on the regular print papers, so the Nature, Science, and so on, one other thing that has become very, very popular is the use of video animations as a supplementary element. Video animations are very, very popular. They're almost always part of PowerPoint presentations that people make, and very, very often they're in the supplementary materials to better illustrate what the story is that the paper is talking about. I'm sorry for the quality of the rendering might not be perfect on your side, I can see it reasonably well here, but it is lagging a little bit in comparison to normal. You will see it in a moment in the live demonstration after Patrina presents her part. So these video animations are a crucial part of most presentations and of a lot of the regular publications in the regular journals and also in the preprints. And of course, for social media in general, it is one of the most efficient ways to attract the attention of your audience is to have an appealing way of displaying your data. So the videos help a lot. This is one such video. Here's another one. So these two videos that I just shown here have been created with the light sheet that is currently commercialized or already commercialized by Mizar. Is the Mizar tilt. And Patrina maybe can add a little bit more information about it later. But it is a type of light sheet fluorescent microscopy. And in this case, we see a C. elegans roundworm and undergoing a few rounds of cell division. And the video illustrates that we can actually track and display the tracks of this cell division. So the embryo is able to go through several rounds of cell division without any apparent problem or phototoxicity, which is great and is what is expected from this kind of microscope. So in the recent years, we've had a paradigm shift, and we continue to experience that with these three themes, really. One is light sheet, which I already mentioned, allows for both high spatial and temporal resolution imaging at a reasonable cost, and it is not hard to set up. In addition to that, we have also tissue clearing techniques that have made it possible to image extremely large samples, and expansion microscopy that lets regular people image at super resolution. So these three techniques together have made it possible to image bigger, deeper, and faster. And so the data sets are enormous and often very, very complex. So it's important to have a good grasp on the technique you're using, not just the imaging, but the sample preparation, and then the downstream part, which is how to visualize and how to present the data. So with that, I'll pass it over to Patrina, and she will do the next section, which will include an example of how to prepare data for presentation.
Okay, great. Thanks again, everybody, for joining in. We just got a really great overview of the history of microscopy and a little bit of how we've used microscopy images to communicate our scientific findings. So for my part of the webinar, I'm going to focus on the practical sides and go over some pro tips for you when you're actually acquiring data and then post-processing that data to get your images ready to start communicating your scientific findings. So I've spent a lot of time sitting in front of a microscope and post-processing images to help people understand what they're looking at. And many of these images have gone on to be in publications, presentations, grant applications, etc. So I'm really excited to share with you my process for doing this. I'll start by saying that scientific communication is a really big deal. It's not just important for the researcher and their career and their funding, but also for science in general and the future of science. So it's our responsibility as scientists to communicate our findings not only to our peers, but people outside of our field, to our grandmas, and to the public in general. So this is something that should be on all of our minds. So what I'm going to do for the next few minutes is give you a set of pro tips on the acquisition side, so when you're sitting in front of the microscope, and then also on the post-processing side and how you can get these images ready for presentation. So these are the steps that I like to take, and there are many different ways to do this. And so a lot of these are very basic, and if you're a microscopy expert yourself, I encourage you to share your tips as well. There's a lot of people out there that could really benefit from knowing your thought process as well. Okay, so the first tip that I'd like to share is about always keeping your end goal in mind. So when you're grabbing a data set or capturing some microscopy images, always keep in mind what you're going to do with that. So what is it exactly that you want to show, and where will you be presenting this data, and in which format? So are you going to be giving a talk at your next departmental meeting, or maybe at a conference? Maybe you need some images to pop into a poster presentation for a conference. Perhaps you're a grad student that has a committee meeting coming up, or maybe you're a PI that has your tenure committee meeting coming up. Maybe you need images for that, or maybe you're going to put it into a grant application or a publication. Perhaps you might be wanting to put some nice images onto your website to help recruit grad students or postdocs. Or maybe you just want to impress your friends and your science friends and put up a nice Facebook cover. Whatever your goal is fine. Just make sure you know that ahead of time. That's going to help you decide how to set up your experiment and help make you more efficient as well. Okay, so the bottom of this slide is listing the 3 main tips I'll go over. These top 2 bullet points are things that I want to mention but are outside of the scope of this webinar. So you want to make sure you always have a good sample to start with. So make sure you have your sample prep down. The saying goes, garbage in equals garbage out, and that's especially true for microscopy images. So if you start with a bad sample and you get bad images, you're going to have a hard time working those up, and they're not going to be very helpful to help communicate your findings. Also, make sure you have good experimental design. So make sure you know what your microscope is capable, you know the emission filter sets, you've got your controls set out. Okay, so what we're going to spend a few more minutes on are these bottom 3 topics. So you want to always max out your signal to noise, and I'm going to show you or talk about some ways to think about that when you're in front of the microscope. And then I'll show you a great example of why you should always be not be greedy when you're taking microscopy images and really focus on only acquiring what you actually need. And then lastly, I'll try not to lecture you too much about making sure you have all your metadata. So this is going to be information about your imaging and your images that you will need later on. Okay. So let's say you have a great sample, your sample prep is all down, you've got great experimental design, now you're going to go sit in front of the microscope. The first thing you should be thinking about or make sure you know is the dynamic range of your detector. So this is usually expressed as a bit depth. A really common one is a 16-bit camera. So this means that it saturates at 65,000 something gray level counts. This is going to be important to make sure you don't saturate your detector, as well as to help you read your image histogram. So on the left, I'm showing an image histogram that I created in ImageJ. And what you're looking at here are the number of pixels on the y-axis versus their intensity or their gray levels. And then below that, it's showing some basic statistics about this histogram. So I want to point out the min and max. So this is the dimmest pixel in your image and the brightest pixel in your image. This is the signal to noise ratio that you want to pay attention to when you're setting up your experimental parameters. So try to get this as high as possible or as high as your sample will allow. So this is where you'll want to adjust your exposure time, your laser power, your pinhole size, etc., to really max out this ratio. So when you see these really stunning or striking images that are oftentimes on the cover or going viral on Twitter, these have the signal to noise maxed out or are very high. It's hard to put an exact number on the ratio that you should go for because it's so sample dependent, but you should just try to get this as high as your sample will allow. Okay, so a little side note on maxing out your signal to noise. You want to pay attention to signal to background, or what I sometimes hear called local signal to noise. So let's take a look at this image on the right. So if we look at the pixel or gray level intensity or gray levels or intensity count of a random pixel in the background, we see that this is about 100 counts. So this is basically your camera noise. Now, if we look at one of these brighter structures in the nuclei here, we see that the max counts there, or the counts are approximately around 450. So I'll see a lot of people look at this min and max of their histogram and say, oh, my ratio is 4:1 or better than 4:1. But what they actually, or what you should be paying attention to, is your local signal to noise. So you can use a pixel inspector tool in your image acquisition software and start scrolling around and getting an idea of what the local background to noise signal to noise is here. So in this particular example, if we look adjacent to the nucleus, we see that these pixel counts are around 240. So this is showing that our local signal to noise or signal to background is actually only around about 2 to 1. So this is, all my examples are in image J here, and I'm just highlighting up here that if you scroll over any of the pixels in your image, it's going to show you those values up here. And any image acquisition program will show you both the pixel counts or intensities for individual pixels, as well as the histogram that I showed you on the last slide. So these local signal to noise become very important later on, or can be very important later on if you're doing certain post-processing steps such as deconvolution. So again, you really want to think about maxing out this signal to background ratio to try to get the best possible quality image. Okay, number 2. I call this take only what you need. So this is advice to think about exactly what you're trying to accomplish and to also keep in mind what you're going to be doing with these images downstream. So here on the left, I'm showing you an image of this tardigrade that I took. And this is actually a movie, a long time lapse of this tardigrade moving around. And when I went home to process this data off of my laptop, I was really frustrated that instead of just cropping this image, I took this very large data set that resulted in a big file size that was hard for me to post-process later on. So my point is to think about what you want to do with this, what you really need. So in this case, we don't need all this black stuff or these black pixels, and how you'll be processing the data later on. So if you're just working on a laptop with limited RAM, keep this in mind. It'll make your life a lot easier later on. My last tip on the acquisition side is to make sure that you have all your metadata recorded in a way that you understand. So most image acquisition programs are going to have all that metadata captured, but make sure you understand critical things like which objective you're using, the camera pixel size. You're going to need this later on to put on timestamps and scale bars. So if you don't know how the metadata is really saved or you don't understand it, make sure you take very good notes for that. And I'll talk a little bit more about that in the case study shortly. Okay, so those are my main big tips for when you're sitting down in front of the microscope. I really can't emphasize how important it is to look at your image histogram. I see a lot of people on microscopes just adjusting things and they're not paying attention to this, and so it seems like they're arbitrarily changing things. Okay, so now let's transition to pro tips for getting your images presentation ready. So keep in mind your end results or what you want to do with this and what the final requirements are for this, and I'll talk a little bit about that more shortly. And then I'll go over an example workflow with you that goes over my thought process for how I like to think about working these images up. And the last thing I want to mention is to make sure you report everything that you do to your images and don't ever do anything that's misleading. Just show what the images show or show what helps support your scientific findings or your other quantifications. Okay, so I'm showing this slide again because this is absolutely critical for your post-processing. So always keep in mind what you want to do with your data, and importantly for this step is where you're going to publish it. So this becomes incredibly important. For example, if you're going to put it in a poster, there might be certain file format requirements, or if you're going to post it onto social media. So for example, if you're going to put a movie up on Instagram, you should know that Instagram doesn't allow movies that are longer than 1 minute, and it doesn't accept GIFs either. So this is where you want to start thinking about these final details of where you're going to publish. And then make sure you start post-processing your data in a way that makes that easier. Okay, so here's our example workflow. So what I have is an image, a Z stack. Here we're just looking at a single slice of a C. elegans embryo that's labeled with H2B RFP. So in this example, we're going to go to a mitosis conference, and we really want to highlight this dividing cell in our poster presentation. So the next steps, what I'm going to do for the next steps is show you how I would start processing this data to make it look really nice to pop into a poster. Okay, so step 1 is that you'll want to adjust your scaling. So you'll put your image into some kind of image processing software. You can use something like Fiji or Elements. It doesn't matter which one you use. You can do all of these steps in any image processing software. So in this case, I loaded this image into Fiji. And the first thing that I see is that it's completely black. And so what you'll want to keep in mind is that this is scaling to the dynamic range of your detector. So if we look at this image histogram, we see it's going from 0 to 65,000 something. So most fluorescence images, if not all of them, are actually using a very small portion of or range of the dynamic range of the camera. So the first thing you'll want to do is auto scale to your image. And most softwares have this ability to do this transformation for you automatically called auto scale. In Image J, it's this auto button on the brightness and contrast window. I'm pointing this out because I can't even tell you how many calls I receive where people think something is wrong with the data and it didn't transfer properly. So make sure the first step that you do is always adjust your scaling. So when I press this auto button, I get an image like this. And so this is the one that I just showed you previously. So what we're doing here, if we look at the histogram now, is all black pixels below 82, or any pixel intensity below 82 is black. And then the pixels with the count of 461 are now showing up as white. And so now we can see our dividing our C. elegans embryo here. And what you'll want to do is play around with these different minimum and maximum sliders to get it to a contrast that you would really like. I would definitely advise to not go overboard here. Sometimes I see people pulling up the minimum counts really, really high and clipping a lot of the data so it looks really black on the background. You'll want to be cautious doing that on the maximum side too, because you can start clipping a lot of your data because all of those pixels will appear to be white. Also keep in mind, for example, if you're presenting on a projector and the quality of that projector. So I'm sure you've all been to one of these presentations where you can't see any of the images. So in my experience, what works well is to actually not threshold too much on the bottom. So keep some more of the gray pixels and then make it extra bright or do a little bit more scaling on the maximum side. And this tends to look a little bit better on those kind of crappy projectors that you see out there. Okay, so step one, make sure you get your scaling adjusted how you like it. And then step two is what I'm calling find the best view of your data. So this means what kind of what's the best way to show what we're trying to focus on? So as I mentioned before, this data set of a C. elegans embryo is actually a Z stack. So it has several different Z slices. And what I've done here is a maximum intensity projection of that data. Another option might be a volume rendering, or maybe I think in this case, it's better to just show a single Z slice because we really want to highlight that dividing cell. And in my opinion here, when we do the maximum intensity projection, it looks a little too cluttered and it's hard to see those chromosomes pulling apart. So for this example, I'm going to go ahead and just do the single slice view. One of my pro tips would be, and something I always do on all Z-stack data or 3D data sets, is do a maximum intensity projection to quickly look at all that data and kind of get an idea of what I'm starting with. Step three, I'm calling post-processing, and this is completely optional. Some people really don't like to do any post-processing, so you do have the choice of just leaving your image in its original format. If you look into Image J on the Processing tab, there are many, many different transformations you can do. Here's one that's called smoothing, so you can see the difference from the raw data on the top panel. You can do a ton of different things, different transformations to your images. Another option is doing deconvolution. So here I've deconvolved with a plugin, the Microvolution plugin for Image J. So you can see we get a nice little boost in contrast here. Whatever transformations you do to your image, that's fine, but you need to make sure that you accurately report that. And this can be reported on the image itself or in the figure caption, and then please note that you should be performing any image analysis on the original data, not the processed data. Okay, step four, this is one of my favorites, and I'll admit that sometimes when I'm avoiding my real work, I'll just play with images and change their lookup tables. So this is where we get to pick the colors of our image. And so in general, definitely for publications and whatnot, grayscale should be your default. This is very easy for the human eye to see lots of variations, but sometimes we can use a little bit of help with the lookup table to help see dimmer structures. So I'm just showing you 3 different lookup table options for you. These are some of my favorite, and I think inverted is especially nice for dim structures. Also, it helps saving on if you're printing, so you don't have to print with so much black ink. Be mindful of colorblind people when you're displaying multiple colored images. Try not to use green and red, that's harder for them to see. And I definitely recommend checking out this paper by Jamie Johnson that goes over, how you should be transforming your images and what lookup tables you should be using in the context of how the human eye can see. So it's a quick read and something that's, I think, pretty interesting to check out. Okay, so step 5 is adding your scale bar and your timestamp. You need to tell people how big the thing is that they're looking at, or how big your image is, and also how fast you're imaging if you have a movie. I think this can be a little bit misleading if you don't present this information, and this is why it's so critical to make sure you have your metadata captured properly. So if you're going on later and you don't have this information, it'll be hard for you to add a scale bar or a timestamp. So you can do both of these things in all image processing softwares, especially like Fiji. Another option, especially if you keep track of your metadata, is that you can add a scale bar later on in something like PowerPoint. This is a good option if you want to make have more freedom on how your scale bar looks. Okay, so the last step in this workflow is to export your data and finalize. So as I pointed out a few slides back, you want to make sure you know the format that's acceptable for the medium that you're publishing in. So if it needs to be a JPEG or a PNG, make sure you're not exporting as a TIFF, for example. Then you want to label everything as necessary. So in this case, I'm indicating that this image has been deconvolved, that it is just looking at a single Z slice with the number here. This was imaged by Dave Mattis in the embryology course. And I'll emphasize again to make sure that you report everything you do. So in the figure caption, on the image like I'm showing you here, and also I'll advise to not let your biases come through and don't mislead people. And there was a really great guide for beginners that just came out recently. I'm giving the reference here, and I highly, highly recommend that you check that out. Okay, so that's all. Those are my 2 sets of pro tips for you for when you're sitting in front of the microscope. Definitely go for signal to noise and check out your image histogram. As I mentioned, there are so many people that don't even pay attention to this. Also make sure to have your metadata. And then lastly, think about your end goal in mind for your post-processing and make your workflow a lot easier, and be sure to report everything. And then the last slide I'll show you here is just my contact information. I probably think about this stuff in an amount that would be considered unhealthy, but go ahead and definitely reach out to me. I love talking about it. I love helping people. So you can email me or call me or text me. And then I'm always on social media posting a lot of non-nerdy stuff. So follow me on Twitter and LinkedIn. And then also go ahead and check out the Mizar accounts. We're posting a lot of cool stuff there. And thanks, Luciano, for having me. And now we get to see the really cool cutting edge stuff from Luciano.
So thank you very much, Patrina. I really appreciate your input on this, and I think everybody here really benefits from your pro tips. And I do encourage you to follow her on Twitter. She is posting quite a lot of interesting material, sometimes pro tips as well. So when you do capture an image like that, a 3D image, one of the things you may want to do is do volume rendering. So this is essentially taking each of the slices, the 2D slices that you capture are made up of pixels, and these pixels have a depth which makes them a 3D pixel, so a voxel. And these voxels all have an XYZ position, and time, of course, if it's a time lapse. But they have an XYZ position, and with volume rendering, you're plotting these in the 3D space, and then assigning different intensities or different values, gray level values, based on how your image was acquired. You can image at pretty high spatial resolution. This is an example And you can have extremely large data sets. The case is that some of the data sets I've seen can be hundreds of gigabytes in size. And if you have large data sets, you need specialized software that will handle those. And the tool or the technology that we use is a multi-resolution, multi-block approach, which essentially in the background breaks up the data set into multiple blocks of different resolutions and lets you experience the whole data set on a laptop, like I'm using a laptop right now. So it lets you render an image that is much, much greater than the amount of RAM that you have available. So volume rendering often looks like this. So gray level, that's what you saw also on Patrina's slides a moment ago. You can change which intensity values are mapped to a particular color, and that's what a lookup table is. You're essentially mapping an intensity of 5 to a color, to a particular color, and then of 6 to another color, of 7 to another color, and so on. In reality, it's more like a range between, say, 10 and 100. You have one color. Between 100 and 110, you have another color. And in many tools, you can actually customize these lookup tables. So that's what a customized lookup table looks like right here. If you zoom in, it looks like the figure C. And if you zoom in and you have a surface already generated, it looks like D. So here you have volume and surface rendering. Even though the surface rendering in this case is not full surface, it's just the triangles that make up the surfaces. And on the top right, you see a full surface, and each of the little nuclei there, or the cells, have been colored with a different color just so that it's easier to see. This is how surface rendering is created. It's created by applying, in the simplest way, it's applying a threshold on a different level of intensity. And if you do that at different Z positions, then you can have a thresholded volume, which would look like this. And then you can create a mesh that connects, or a sheet that connects all of the voxels of the same intensity or similar intensity, making then a continuous sheet around the object of interest. So why do people bother even doing surface rendering? Well, there's a few reasons. Some of them are relevant to this presentation, and these are the ones on the bottom. One is you can color them differently, completely differently from the original data. And this sometimes is useful in case you want to show, for example, the volume as a color. So small objects will have a particular color, big object will have a different color. Or if they're moving, you can color code them based on the speed at which they're moving. Also, you use a lot less memory. Once you save the file as a surface, you can render it as a surface, and that's typically a lot less data than if you do it in volume. And this is for the example we had. It was 190 megabytes, that particular small data set, and it went down to 60 megabytes for the same data set. And it's a lot easier to interpret because now we have very clear boundaries with black background. And that's another reason why sometimes people like to use that. So in Aivia, this is what it looks like. You have a big window with all the data. Here is where we have all the channels that you may have acquired, and then the objects that you've done the analysis on. You can change the types of rendering, and you can adjust the maximum and minimum that is displayed on the screen. All of that is available, and I will show that in a live demo in a moment. So that's displaying things on a normal computer screen or on a video projector. But you may want to do this also using augmented reality or virtual reality. So one approach that I sometimes use is using a very simple, very interesting app called Sketchfab. It looks like this. So let me see. I have here. We have an account on Sketchfab as well. And yeah, so what I'll show you now has been generated in Aivia, and we uploaded the model to Sketchfab so that we can then render it in VR using a simple headset, like those Google Cardboards that you've for sure seen around. And that means if you have this in one of these Google Cardboards, you can visualize this in VR very simply. This, you can turn it back on. This is also interesting because you can actually embed these, this is a website, so you can embed this website with all the interactivity that you see there, so the 3D rendering and interaction on any website. There's just a share button and you can share that directly on your own website, on your social media, really anywhere. So that's a pretty easy way to use VR or AR. The other approach is using software that is dedicated for this purpose, like Aivia. And in that case, you have the software running on your computer, you have a headset, one of the more high resolution headsets on, you press the button, I'll show you that in a moment, and then you can dive into your data and explore it. And there's a lot more interactivity, of course, because you have controllers, they have lots of buttons, you can grab things, move around. It's a lot better, of course. But those are the two options for virtual reality. For augmented reality, one of the apps that I've used in the past, apart from Sketchfab, is this one called Augment. And this particular one, I can show you what that looks like. So this researcher has made a model of the NPC, so the nuclear pore complex, and he has that rendered on top of his real poster. So he's at the conference, he has a poster on the wall, and he can point his phone at the poster, and on the phone he can show people that are watching the poster, looking at the poster, he can show this little animation of the nuclear pore that he has created himself. And you can put there basically anything you like on that application. We've done this as well. We have it here on Sketchfab, and you can find it also on Augment. We used this at Neuroscience last year, and it was a lot of fun. So some tips about image visualization. So make sure you're using the right hardware and software for the purpose. If you're using Aivia, it helps a lot to have fast SSD drives and a good NVIDIA GPU. Another thing that Patrina also mentioned is to have the metadata to hand. And so make sure that once you load the image into your software, that the parameters are correct in relation to what the acquisition parameters were. Use the dynamic range in a smart way. And if you're acquiring, make sure that you have data on all of your on the whole of the dynamic range. Don't have big areas with nothing in there, because it's a bit of a waste of space in terms of disk space. The lookup tables can be your friend, as you saw. Patrina also showed that, but they can be your enemy. So don't get lost in creating endless lookup tables. Better optimize one and make sure it works well to highlight what you want to show in your story. And choose the right type of volume or surface rendering. In Aivia you have a couple, but you could have even more in other tools, like in Blender or in other tools there are even more. And then there's some advanced tools for display and data exploration that can be very useful, like the orthoslicer or a clipping plane. And finally, remember where you're presenting this. So what kind of media is this going to be on? Is it on a poster? Is it a presentation? Or VR and AR? So depending on what you're aiming for, you have to have a little bit of consideration on how you prepare the data. So this is one of the data sets I used to introduce this webinar. This is a rabbit that has been imaged with a technique called optical projection tomography during a EMBL microscopy course on developmental biology that is organized every year near Lisbon in Portugal. The author is Gabriel Martins, and you can find him quite easily online. So this data set is currently displayed with volume rendering. And up here you have this histogram that Patrina showed, and you can control which voxels are present. So I can control here which ones are present, and I can increase the contrast and the brightness of them. This is just for display. It doesn't actually change the intensity of the data. You can change the volume rendering mode, and that's what I'll do. Adjust it. So this is a composite mode. It's one of my favorites to display data. And you can use a custom lookup table. Okay, so in terms of visualization, these are the first few things I would do every time, which is to adjust the histogram so that I'm highlighting the features that I'm interested in. So if I was interested in blood vessels, I would probably leave it like this. If I was interested in looking at the actual structure of the whole rabbit, I would leave it more like that. And then if I wanted to explore the insides of it, I could use an ortho plane over here to cut through the data. So this is cutting through the data orthogonally. It is possible to place it in any angle, like this. And by the way, in a few moments, I'll also show how to animate these slicers to make videos that incorporate all of these tools, or a clipping plane which will cut away part of the data. So that might be something you also might want to use if you want to highlight or hide part of the data. And you can use them in combination, multiple clipping planes in combination, in case you want to cut away a part of it. So right now, oops, too fast. We have the face of the rabbit can be partially removed so we can look inside. So these are some of the visualization options that are available in Aivia. And if you have multiple channels, then the channels will all appear in a list here. You can also represent this, instead of volume rendering, you can do it with surface rendering, and that's what it would look like. The color of it can be changed to any color you like, and you can use this in combination with the volume rendering. I think this is one of the things I showed in the video that we posted online, is the surface going through the sample. Okay, so this is a mix between surface and volume rendering. These are just some of the options you have for preparing your data to display it. And sometimes it's even useful to do the presentation or do part of the presentation in the software itself, because you have so many more options and you can answer questions directly. If somebody asks something specific about the data set, you can quickly navigate through it and show it live. So if you go now into the second or the last part, which is using the visualization, but going one step beyond that, and it's about creating animations from this visualization, how to create those, and what can we propose as good rules or good tips to use when creating one. So before we get into this, I'd like to show you this brief video. It's a minute. I find it really cool. It has there's three clips. They look the same, or actually the story is similar. It's about group work. And hopefully this isn't too slow on your side, but there's a group of penguins and they're going to one side, and yeah. So they managed to escape the shark there. So I'm showing this as an example of a short, very, very short movie that actually has all of the key components of a good movie. All of these 3 short movies that I'm showing have that. And these are, the components are the setting, the characters, the plot, the problem, and then the resolution. So with all of these, they are covered, and they do a really good job at that. I think this was actually an advert to some sort of bus system in a country. But so the 5 elements, in our case, we have the setting, and typically that is our microscopy sample, whatever that may be, and that's why we always describe it and say, okay, this is a C. elegans, or this is a rabbit in my case, or this is whatever it is. Explain what the setting is, where this is happening. Then the characters, which may be a group of cells, or it may be a single cell, or even an organelle inside a cell, or a group of organelles. So describe those and introduce those as characters of your movie animation. Then there is a plot. What happens? What actually happens in this video that you're creating? What's the story? And then it helps a lot to have a conflict or to have a problem. This is what captivates, typically, this is what captivates the audience. And often this is put at the very beginning of a video. Even outside of the video, it's just introduced by the presenter, sometimes as a hypothesis, but sometimes it's just a problem they want to solve. And then, of course, during the video or just after the video, there should be some sort of resolution that brings closure to the conflict, and that these 5 elements make a very good, if you can cover all these 5 elements within your 30-second video, that works really, really well. Another way to look at this is the Freitag pyramid, or in this case, the extended Freitag pyramid, which is a little bit, the pyramid typically is just the middle part. In this case, there is a resolution and exposition. And this is what it would map to what we do to science. We'd have the hypothesis and introduction. That's what is often called the exposition. Then the rising action are the results, as we discuss the results. And this would be the whole presentation, of course. This timeline here is the whole presentation. Then the final result, or the key result, would be the climax, and then a discussion which is the falling action, and a conclusion at the very end. So you can also try to fit all of this into a video. It's a bit more challenging. Often people will have little videos that will help tell the story of each of these results, or at least of the key results. And that's typically how videos are used, rather than to completely take over the whole story. So how are these videos actually created? This is the only technical piece that I'm going to talk about. We use keyframes, and that's the same concept that you have with these books, video books. You probably all tried these when you were small, or if you have kids, you might have tried it recently. And these videos or these books have still pictures, of course. But if you pass them very quickly, it looks like there is a moving figure there. And that's because our brain is interpreting or interpolating what is happening between one page and the next page. And that's essentially what we have to do also with software, is take keyframes that the user defines and interpolate the movement from one to the other. And that creates a sense of motion. So what does that look like? For us, it looks like this. So it has a strip at the bottom that you can add keyframes to, and you can add as many as you like while changing either the angle or the what's on and off. So whether you have a surface on or a surface off, volume on or off, the clipping planes and the ortho slicers, all of that can be controlled and is saved into the keyframe. And then we interpolate the motion between one keyframe and the other keyframe. So I'm just about to finish there, and then I'll play it. That's the last one. Then I'm going to play that short movie. And that's what it looks like. So this is already interpolated. It's going to all of the keyframes that were added, and then you can export it. So some tips before I do a brief demonstration. It helps a lot to think about those 5 elements. You might not always use all 5 elements and really introduce them into your video, but it's worth thinking about them and deciding whether or not you want to keep them in the video. One that I never take out is what the problem is or what the conflict is, because that is normally what drives the story. If you have a long video, then it helps a lot to do a storyboard. Either just sketch it out with pictures or just statements. In this keyframe, I want to show this, next keyframe, I want to show that, and have the key concepts of your video written down so that you remember to capture enough keyframes to show your full story. Well, I suggest that you keep the camera movement as constant as possible. So don't accelerate and slow down a lot, because that tends to make people seasick. And if you are seasick, definitely don't zoom in and zoom out too much, because that makes it very hard actually to watch the movie. And typically I start with a quick draft and then add more frames or edit the frames that we already have available. So you can do that and create the perfect video. So I'll get back here. So to start it, just turn on the video animator at the bottom. Just reset this whole scene. So I'll start my first frame. I want it to be in this position. So I'll just add a frame here. So I don't want the box. So I add a frame, and then I'm going to rotate him, add another frame, zoom in, and rotate, add another frame. So this is already a simple animation that we have right here. Now I'm going to introduce one of these other tools, for example, the clipping plane that is at the back there. That currently isn't cutting this. Okay, now it is. So I can add it and do that. And so that will now have this transition that will add the head that was just gone. You can also add the ortho slicer that was at the bottom. You can start it up there, lower it down, and you can follow it as it goes down and come down with it. And now that portion is also there. So if I play the whole thing now, it will go through all of those transitions. As you see. Then I can introduce additional elements. So I'm going to add here the blood vessels, and I'll have a new clipping plane that will strip away the volume and leave the segmented results. So I'll start it here, rotate it, and then gradually move it over And that's this last portion. So it's slowly stripping things away and adjusting. If you wanted to edit any of this, it's very easy. For example, let's say this isn't perfect because maybe you want to have the full rabbit visible. So you can zoom out a little bit and just go to the frame that you wanted to edit and click on the camera, and that will adjust that keyframe. So now there is a zoom out and a transition between the two, and then it will zoom back in, of course. Okay. Then the next part could be important for your department, the branding part. So by default, you get the Aivia logo. You may want to add some other logo. Let me see if I have here. There it is. And you can define how big it is. So I'm going to set it to large, and you can see it on the top. And then choose what kind of resolution you want it at and the frame rate. And then save. This is going to then save it somewhere on your desktop or somewhere where you would like to have it. So let me. And it's now starting that process. So it hides away the video as it records. And once it records, then you can watch it. I'll leave it recording. It may not finish while we are here. It takes a moment to render the whole thing at high resolution. So that was the video animation demonstration using the ortho slicers and the clipping planes. There's even more options, but for today, I think that's where we'll finish. I wanted to conclude, and of course, I'd like to ask Patrina also to have final remarks if she has anything else to add. But as a final conclusion, I'd like to say that you should always think about the audience first. Who are you going to be talking with? As Patrina said earlier, you should think about what level of information you want to provide, the details, and also the angle that you're using. And after that, create the content that is appropriate for that audience. Have a story that is compelling, and quite often in science, the stories are very compelling and very interesting if they're presented that way. And pick the right tools and know them well so that you can make good use of what you have in your hand. Whether you're using ImageJ or using Aivia, there are very good tools out there. And finally, be creative. Don't be, especially if you're using these tools to create material for a oral presentation, you can be very creative or for social media. Of course, if you're presenting or preparing materials for grant application or for a publication, it has to be a little bit more, or typically has to be a little bit more conservative. But being creative certainly helps to stand out and to show, highlight your story in a more attractive way. I wanted just to go back to Patrina for a moment in case she wanted to add anything else before we go to the questions.
No, I think you covered that well. I'd say thanks for joining again, and definitely reach out if you want to share or have any questions.
So I would like to thank everybody for joining. If you have any questions for us, either me or Patrina, feel free to contact us via social media or any other channel that you prefer. And I hope to see you again next time for the next webinar, which will probably be in a couple of months. Thank you.